let us understand about lipid. Lipid is a biomolecule. Lipid is a biomolecule that we already know, right? And one point you need to remember in here is it is strictly not a polymer. It is strictly not a polymer. That is a PYQ. Okay, strictly not a polymer. Then, if you say about the characteristic features of these lipids, we can write they are waxy, they are greasy, they are oily substances. They are oily substances. Okay, and they are not soluble in water. They are not soluble in water. And in which they are soluble? They will be soluble in what? They will be soluble in non-polar solvents. Non-polar solvents. If we say an example like benzene, chloroforms are example for this. In these they will soluble. Okay. Then if we say about the types of lipids, there are different types of lipids present. First we can say simple lipids. Then we can say derived lipids. Then we can say conjugated lipids. So these are the three types of lipids: simple lipids, conjugated lipids, and derived lipids. Okay. Now, the simple lipids we can say as fatty acid, and glycerol, and we can say glycerol plus fatty acid. Okay. Now, the fatty acid. If you say about the fatty acid can be different types: saturated fatty acid, unsaturated fatty acid. So saturated means single bond in between the carbon compounds. Single bond will be present. Then unsaturated means double bond will be there. Double bond is present. Okay, that is unsaturated. So if we say the example for the saturated, which and all will be the example for saturated, here we can write, right? So here we can write the saturated and unsaturated. So saturated means single bond, whereas unsaturated means double bond. Okay. Then in saturated, the example for saturated fatty acid, first we can write palmitic acid. Then we can write stearic acid. Then we can write arachidic acid. In the case of unsaturated, oleic acid. Then secondly, linoleic acid. Third one, linolenic acid. And fourthly we can say arachidonic acid. Now if we take the structure of glycerol, it will be, the structural formula for the glycerol will be C3H8O3. Okay, so 3 carbon, number 1, 2 and 3, and for the valency we are adding hydrogen. Okay, and 3 OH group, 1 2 3. So this one is trihydroxy propane. Okay, now the structure for the fatty acid will be like a long chain. So fatty acid structure will be a long chain, that is a long chain of hydrocarbons with the one acidic group. Which acidic group? That is COOH, carboxylic acid. Now the fatty acid will be like this: CH3, CH2, etc., CH2, like that, lastly COOH. So this much of carbon we can say it as R group. Okay, so the maximum 19 carbon can occur in the R group. Maximum 19 carbon can occur. So if we say here the palmitic acid, it is a 16 carbon compound. Including the carboxylic acid. So R group will have 15. So we can write R group will have 15 carbon, and the acidic group 1 carbon, altogether 16 carbon. And stearic acid consists 18 carbon. That means R group contains 17 carbon and the acidic group contains 1 carbon. In the case of arachidic acid, it contains 20 carbon. In that, R group contains 19 carbon, whereas it is associated with the carboxylic acid, so acid is 1 carbon. So altogether 20 carbon. Okay, and here I told you in the unsaturated, double bond will be present. In the oleic acid, 18 carbon is present with 1 double bond. Here linoleic acid consists 18 carbon with 2 double bond. Linolenic acid contain 18 carbon with 3 double bond. Arachidic acid also contain 20 carbon with 4 double bond. Okay. Now let us understand the structure for the palmitic acid. So palmitic acid, you know that 16 carbon compound. It is a 16 carbon compound. So the structural formula for the fatty acid is CH3 CH2 NCOOH. So here including the carboxylic acid, here we can write including carboxylic carbon number, it is 16. Okay, so R group consists 15 carbon, acid consists 1 carbon. So we can write the formula as CH3 CH2 N will be 14 plus 1 carbon 15 plus the COH, that is 15 and 16. So 16 carbon. So we can write CH3, CH2, CH3, CH2, 14, COOH. What about the stearic acid? Stearic acid. Stearic acid. So stearic acid will be containing 18 carbon. So we can write CH3, CH2, 16, COOH. How we will write the arachidic acid? Arachidic acid. How you will write arachidic? Arachidic acid is 20 carbon. So maximum number of R consists 19 carbon and 1 acid carbon. That is carboxylic acid carbon, right? So it will be like CH3, CH2, 18, COOH. This will be the structure, right? Now the glycerol plus fatty acid. I told you there are different types of simple lipids. They are fatty acid, glycerol plus fatty acid. So in the case of glycerol plus fatty acid, three types are there. One is monoglyceride. Second one, diglycerol or diglyceride, and thirdly we can say triglyceride. Okay, now the monoglyceride means one glycerol plus one fatty acid, whereas diglycerol means one glycerol plus two fatty acid, whereas triglyceride means one glycerol plus three fatty acid. Clear? Monoglyceride, in monoglyceride, diglyceride, triglyceride, the glycerol number will be same, that will be 1. Okay, in all the glycerol number will be 1. What is changing here? The number of fatty acid connected to it will change. Okay, so first let us draw a monoglyceride. So for that we required one glycerol, right? So the glycerol structure will be 3 carbon. In that hydrogen will be attached to each for the valency purpose. Then one hydroxy group will be added, plus to the first carbon the first fatty acid going to attach. That will be, we can write this like OH, HO, C double bond O, R group. This is one fatty acid. Okay, this can be a palmitic, it can be a stearic, it can be arachidonic, it can be any kind of fatty acid. So that's why I told the R1. When it joins here, when it joins to see for first carbon, what happens? From here an OH, from here one H will be removed, that is one water molecule will be released, and they form the bond. That bond is known to be ester bond. And what structure we will obtain? C, then CH2, CH2. Here O, C double bond O, R1. The structure will be like this, right? So here the bond formed is ester bond by giving an water molecule. So now this is known to be monoglycerides. If it is diglyceride, what happens? This OH, in this OH, H will be removed and it will be added the another fatty acid, that is R2. Then it becomes diglycerides. Become diglycerides. Now it become diglycerides. Now in the third position, if another fatty acid attained, that is C double bond O R3, then it will be triglyceride. Triglycerides. Okay, triglycerides. Now that is all about the fatty acid plus glycerol. Now let us move on to the conjugated lipid. In the conjugated lipid, the conjugated lipid, one example is lecithin. What is the example? Lecithin. So lecithin, otherwise known as phosphatidylcholine. Phosphatidylcholine, lecithin. So for that, it requires one glycerol plus a phosphate or phosphoric acid, two fatty acids, and choline. Okay. So glycerol structure, we can draw this glycerol structure. CCC H2 H and H O first fatty acid, second fatty acid. Then listen here, an OH will be there to this OH, to the third position of carbon. The phosphoric acid is adding. The phosphoric acid structure will be OH. Now the ester bond will take place here. Okay, ester bond will take place here. So from here one H and from here OH will be moved. So one H2O will be released. So what happens? Here? If the H is moved from here, OH also moved from here, right? So the ester bond formed here will be like this. Okay. Then the next structure will be the choline. Choline structure will be CH2OH N CH2 CH3. Okay, this will be the structure for choline, that is CH3 3 and CH2 and CH2OH. Okay, so if we add this here, OH CH2, then N, then CH2 N CH3 okay. Now here also one ester bond will be formed. From here one H will be removed. From here one OH will be removed. And here what the bond will be formed? The bond formed here is the ester bond. So this will be the final structure for the lecithin. Okay, so this lecithin, it is a phospholipid. The lecithin is a phospholipid. That is non-lipid part will be phosphate non-lipid part will be phosphate. Okay, so glucose plus, sorry, 1 glycerol plus 2 fatty acid plus 1 phosphate plus the choline constitute the lecithin. Okay, now let us move on to study the next type of lipid, simple lipid, that is derived lipid. Derived lipid, which is an example for derived lipid. Example for derived lipid is cholesterol. Cholesterol. Okay, so for this cholesterol, how many carbon is there? 27 carbon is there. Fused carbon is present. Fused carbon is present, and it has a fused carbon means ringed carbon. Ringed structure. Ringed structure. Now let us draw the structure for the cholesterol. First a 6 carbon ring, then again a 6 carbon ring, then from here a 6 carbon ring, then from here a 5 carbon ring, then from here 1 2 3 4 5 6 CH3 CH3, then here here 1 2 3 4 5 6. CH3 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 CH3, then here also one CH3, then here one OH group. So this will be the structure of cholesterol. Okay, derived lipid. So these are all about the lipid. So thank you.